Right, this lesson is going to detail some of the information about human genetics and the mutations. The idea here is that there are two main types of mutations. We have chromosomal mutations and gene mutations. We have focused a little bit on gene mutations already. We did a lab. We're going to really focus today on chromosomal mutations and then add a little bit at the end with gene. So chromosomes, at first, if we have talked already, humans have 23 pairs. 23 would be our haploid number. And so if you have them in pairs, 46 is our diploid number. The chromosomes are made up of DNA, and under normal conditions, all the chromosomes are inherited, are intact. So you would have a full sequence of DNA, but we're going to talk about what happens when things go awry, and there might be some instances that you don't get a full set and the entire chromosome is not intact. Okay, chromosomal mutations. Any change in the structure or number of chromosomes could be classified as a chromosomal mutation, so we'll go through kind of both of them. And when you affect an entire chromosome, you're really going to affect many genes on there. You can't really predict how many you're going to have, but you're on a larger scale than if you have a gene mutation, which we will discuss in a few seconds. So here are the types that we're going to talk about, and we'll go through each one. For right now, all you really need to recognize is what they look like. In future classes, you might talk about some disorders that are caused by them, but this is just a building course, so let's get the foundation, and then you can kind of elaborate in future years. So the first thing, on the left-hand side right here, we have a chromosome that has a sequence of genes, and all they've done here is labeled them A through G, where A could be any particular gene, such as widow's peak. B gene could be anything like hair color, C gene. So you can kind of assign a specific gene for them, but for this process, you don't really need to understand that. These are just generic letters to represent genes, and look what happens at the end. You have a loss of letters C and D from the original, and so that would be classified as a deletion mutation. So you would just have a much shorter chromosome, and if these would be genes, then this chromosome would be lacking the sequence to produce those proteins, which that's what genes eventually encode for, is a protein. The next one is a duplication. And so here we have the original, once again, A, B, C, D, E, F, G. But then what we see here in the follow-up is that there's a repeated or a duplication of both C and D. So you have a sequence of genes where there's a repeated gene on the chromosome. So that's what you're kind of looking for. And that's the purpose at this level, is just to understand that you can identify a duplication by seeing the end result of the chromosome. So what causes this is actually a chromosome, usually during Meiosis I prophase, forms a tetrad. And if a chromosome crosses over properly, then what you do is exchange gene for gene. So you would potentially, if this chromosome crossed over with a homologous chromosome, then what would happen is, say this line right here is where it crossed over. The homologous chromosome is lined up. You got to imagine that idea. There's a homologous chromosome lined up right next to it. And what would happen if this is where it crossed over, then this F gene and this G gene would cross over to the following chromosome. And then that homologous chromosome would give its F and its G gene. It may be a different variation of that, but you're getting the same gene, but just a different variation, which is good for diversity. But in the case of chromosomal duplication and deletion, imagine if the second chromosome that was homologous lined up unevenly. So say that the second chromosome up here lined up one gene short. So the A gene was here, the B gene was here, C, and so on and so forth. What's going to happen eventually is that if this is where it would cross over, the F gene for this chromosome is located to the left of the crossover line, but the F gene on this chromosome would have been to the right. So therefore, when it crossed over, the F gene would have been brought to this chromatid. So you'd have a repeated F gene. Hopefully that kind of makes sense. In this PowerPoint, it wasn't shown that, but I want to explain how duplication and deletion actually occur. It's a failure of crossing over to happen properly. The next one is an inversion. And what we have, the original has in sequence A, B, C, D, E, F, G. But look what happens in an inversion. You have the same sequence of letters, but what happens is there's a flipping of the genes. So you have A, B, but look at where C, D, E, they kind of inverted or flipped around. So that's kind of what happens with inversion. You have genes, all of the same ones, but just in a different order. And the next one is translocation, and translocation is where crossing over should occur where homologous chromosomes are paired. But in the case where you actually, so this diagram shows you a piece of this chromosome detaches from its original and then reattaches on a non-homologous chromosome. So this green represents a non-homologous chromosome. So if you're kind of doing the same thing that we showed you in previous examples, if A, B, C, D, E, and F were all along this chromosome, this being a non-homologous chromosome might have W, X, Y, and Z. So what will happen is a segment will transfer to a non-homologous chromosome. So the end result is you're potentially lacking letters here, but those letters that you're lacking are now on another chromosome. So now you might have A, B, C, W, X, Y, Z. So what you're looking for is kind of an addition of letters that weren't supposed to be there. And that just means that non-homologous chromosomes might have crossed over, or a piece of a non-homologous chromosome might have transferred to this chromosome. So the last one is what happens if an entire chromosome might be inherited incorrectly. So this process called nondisjunction, that's a key word to understand. It's when chromosomes fail to separate. So during meiosis, this picture shows that at anaphase, this is particularly anaphase 1. Anaphase 1, a tetrad should separate. So this top tetrad separated properly, but here, the tetrad that's below did not separate properly. So therefore, this spindle fiber that was supposed to attach on this side failed to attach. So therefore, when anaphase occurred, all of the chromosomes go to the left. And therefore, that sets up a result where this chromosome, or this cell on the right, which could be a possible sperm cell, is missing an entire chromosome. This chromosome, or this cell over here, has an additional chromosome. So what will happen is, if this sperm becomes the sperm that fertilized the egg, you get an extra chromosome, and that's called trisomy, where this cell is lacking a chromosome because that chromosome that should have been over here is now over in this cell. That's called monosomy. In humans, trisomies happen more often than monosomies because monosomies don't really have the ability to grow in utero or within the development stages of an embryo. A monosome organism is not going to really come to term. They would probably not form, and you wouldn't even really know that it even occurred. Trisomies, there are a few trisomies that occur and will actually be born. Trisomy 21, which is Down syndrome, is probably your most common. But what can happen is nondisjunction can occur at either meiosis 1 or meiosis 2. We're not really going to look at the subtle differences, but just knowing that nondisjunction is the failure of a spindle fiber to attach, and what that causes is sperm or egg cells to have an extra chromosome, and that could be detrimental to the end result of meiosis. So here's what I was just talking about, trisomy and monosomy. They produce gametes, and therefore the end result of a baby with one missing or one extra chromosome. So if this cell were to be the cell that was eventually produced, you would get a trisomy. You get 2 chromosomes from one gamete and then one from what would be assumed to be the appropriate separation of chromosomes from the other gamete. So if these were within a sperm and this was within the egg, this is what typically should happen. You should get one from mom, one from dad. But if the sperm cell inadvertently went through nondisjunction, the sperm cell now has 2 chromosomes, so it unites, and so there's now 3. This case is the monosomy, where this chromosome, which should have been over in here, is no longer present. So when a sperm fertilizes the egg is the only contributing chromosome, thus getting monosomy. So here's a picture that shows nondisjunction once again, more or less in meiosis 2. It just says that there's more opportunities. These 2 gametes are normal. They have 1 chromosome in them. That's what a gamete should have, 1 chromosome. Here, you're going to have one that has no chromosomes. So if it merges with an egg or the other gamete will just provide the one. This one is where you have the extra chromosome. So this gamete would form the trisomy. This gamete will form the monosomy. This gamete will form a normal embryo because you'll get one from this gamete, one from the other. This gamete will also be normal. So here's a picture to show that. So if the egg went through, so if this becomes, right there here, if that is the egg, we have the non-disjunction that occurred. This egg will fertilize with a typical sperm that has one chromosome in it. You're going to get that trisomy, or in this case, what's called trisomic zygote. Just understand trisomy as being the more common term that they identify. So non-disjunction, every cell in the baby's body will thus have how many copies of the chromosome instead of what? The normal is here. And if you have trisomy, so we're going to kind of fill these in. It's just a little review. The condition is called trisomy 21. Individuals have how many copies and of what chromosome? So every cell in the baby's body would have 3 copies of the chromosome. That's what the whole idea of trisomy is. Every cell gets that. From the starting embryo that had 3 cells, from that point on, every cell will thus have 3, but you should have only typically 2. The condition is called trisomy, and trisomy 21 is more commonly referred to as Down syndrome. That's where an individual has 3 copies of chromosome 21. So if you saw trisomy 13, that would indicate that there's 3 copies of chromosome 13. So they kind of just name it based on what chromosome number was the one that you received an extra copy for. Most chromosomal mutations are lethal. So what I said, humans don't really survive very well with having extra chromosomes. The 21 is the most common that can actually survive and reach adulthood. Others will tend to have widespread abnormalities, so therefore, they're not really going to come to term. Trisomy 13 and trisomy 18 are common in terms of chromosomal mutations, but even those abnormalities will rarely survive more than a year. Chromosomal numbers such as 1, 2, 3, and other numbers other than 13, 18, 21, they won't even form. They won't come to term. They wouldn't probably even know that it happened because how quickly the embryo would probably fail to complete the whole process once that point happens. So here's, again, the most common non-disjunction disorder, non-disjunction of chromosome 21. So Down syndrome, I think it's one of those that in textbooks and prior knowledge students have coming into the class. But now that you know, that's what causes it, non-disjunction. So here's a karyotype. We've talked about karyotypes slightly. And so they're in size order. That's how they determine which trisomy you might have, because scientists will know the order of chromosomes. It is size order, largest, all the way down to smallest, with the exception of the sex chromosomes. This would be a karyotype of a female because the 2 sex chromosomes are the same. They're homologous, same size. If this were a male karyotype, the X would be larger than the Y, but a karyotype would designate that. But here you notice there are 3 copies. So non-disjunction occurred, and this karyotype shows a person that would have Down syndrome. We're going to go through amniocentesis in the coming days. So this is just the idea of how do they figure out, how do they get a karyotype? Amniocentesis is that process. So we can come back to that, but I just want to kind of give a precursor of what's to come. Too much or too little DNA, that's what humans, we don't really do very well with having extra chromosomes or lacking chromosomes. So this is just to kind of reiterate that fact. And finally, gene mutations. So gene mutations are when nucleotides or bases are added or changed. So this idea we've talked about, mutations could be point mutations or frame shift. Point is a substitution where we should have an A C arrives. Frame shift is where you actually have a deletion or an insertion. So here there was no thymine, but an thymine squeezes in there. That's going to change one gene. An adenine deletion, that adenine should be there. So here it's a much smaller scale. But gene mutations we've talked about, they just affect really one gene. And here are some examples that we have discussed. So you can kind of read that as your last slide.